Welcoming our first Project Talk speaker, Dr. Karen Leung. She will present on a national workplace navigation hub which empowers student success in the workplace. The first series of her workshop is also listed on the InnovATEBIO website, which we will share that as well. I'm sure Karen will do as well. And so again, welcome to Karen's presentation.
Thank you, Dion. Thank you, everybody, for being here. Excited to talk about our new Workplace Navigation ATE project, which actually just started in October, and will support development and scaling of workplace navigation training, which, as Dion mentioned, we'll be offering starting next Friday. This training was really designed for community college students specifically to effectively assess and navigate any workplace. Want to just share a little bit about the team that I'm working with. So again, I'm Karen Leung. I've been a biotech instructor and internship coordinator at City College of San Francisco for over a decade now. I'm really excited that one of the co-PIs on the project, Naliti Saul, who is the director of the Office of Career and Professional Development at the University of California, San Francisco, is here with me. We also, the two of us, along with many of you may also know James Lewis and Laurence Clement, have been developing this training for over 9 years. And we're really excited in this project to also be bringing in MiraCosta College with our other PI, Barbara Chunkosa and Terry Quenzer. And so I have a short amount of time, but I want to just give you a little bit of background information about how we came to this training for those who may not be familiar with it. I was really fortunate about a decade ago to be able to be part of the bioscience internship program, which had been developed at City College of San Francisco. This program had been developed by James and Laurence, and then further developed by Ying-Soo Lo, with really training in technical skills and employability skills, as many of our programs are, and had really built a beautiful support course to go with that during the work-based learning experience, which was a part-time internship semester long. What maybe is unique or not unique, but just to let you know about this program, students coming into this internship are coming at all levels with as little as 6 units of coursework in biotech before entering the internship program. I've had everything from students who are kind of a semester out from high school to people with PhDs pivoting to different areas of science. And so not only do we have this very wide variety of students coming into this program, we also have them going to a very wide variety of host sites. So everything from academic and government labs, and basically kind of all aspects of science, to small startup companies to multinational companies. So really just it's really exciting. It's part of the reason I love working with community college students, is you get to work with this breadth of students and in all these different areas, but it does pose some challenges. This program had really been thoughtfully designed and continued to be developed there, but there were continually some challenges. I mean, there always are, but I just want to let you know what we were kind of looking at and thinking about trying to address. And so some of the challenges we would have were things like misaligned placements, right? Students weren't necessarily in doing work that they were specifically interested in doing or was going to further their specific career interests. Misaligned expectations. There's maybe not as much communication about what it was that student was going to be doing during that internship, or where they were skill level wise in order to do those particular types of experiments or work. A lot of this stemmed from sometimes students and those supervisors not having great communication across each other. And that not often, but occasionally, because in general there were lots of students very successfully moving into the biotech workforce. But when there were issues often around productivity, morale, and well-being, not just for those students who maybe weren't sure about what was expected of them, or weren't quite engaging effectively with their supervisor, but then the supervisor also feeling burnt out and overworked because they weren't quite sure how to support these students. Most of us all know when we are setting up work-based learning experiences for our students, the students want to do well, the people supporting them want to do well, everybody wants things to go well, and yet still these challenges would arise. And the supervisor would feel terrible if things weren't going well. Occasionally, but not always, in these situations, it might lead to a poor recommendation or not a very strong one. And every once in a while, it would also lead to an early dismissal from an internship, which is obviously devastating for many of us, for us. And then that could lead to loss of future opportunities for students, right? Like if bad situations were happening, then we might not have that host want to host another intern in the future. And so I'm trying not to make this sound dire, but like, what are the issues we were trying to grapple with was like students would be confused and stressed. Supervisors were frustrated. Myself, the internship coordinators, right, were like, we sometimes feel stuck. We're concerned. We're not sure exactly how to help these students. And so we really kind of coming back to that internship program and trying to figure out how to support these students. We recognized, and maybe many of you have as I'm going through that list, how so much of this came down to the relationship between that student and who was supervising them. And that's probably not surprising when we think about our own bosses and supervisors and how important they are to our success. But we realized that really strengthening that relationship, when we saw strong relationships there, internships went very well. Even if we had really well-prepared students and that relationship was not particularly a good working relationship, things would go sideways potentially, right, in unanticipated ways. And so again, about 9 years ago, we partnered City College's biotech program with the Office of Career and Professional Development at UCSF to really develop parallel trainings and resources to support the success in this working relationship. We started by actually thinking about the supervisors and working on developing management training for them to build their strategies and tools to engaging with students. But we quickly realized that even if we were really successful and we got all of our mentors to do this training, our students would go on to other jobs where people had not taken maybe that type of training. And so we realized we really needed to build the parallel training. Our students needed to figure out how to manage up, regardless of where they went. And that they needed additional skills to do that. And we call that our workplace navigation training. To sum up, like the last 9 years, we're now in our third ATE project. We've had fantastic ATE projects to really build out these trainings and provide them to very large swaths of scientists from over 100 organizations and over 300 students from a variety of community colleges. And we're really excited with this project to really, again, scale and not only, I'll talk about a little bit later, have these as virtual offerings that are synchronous, but move to an asynchronous format where people can take this training and put it into their programs as they see fit and aligns with the timing. Because I'm sure many of you know, right, like all of our internships and work-based learning, sometimes even for each of our individual students, happens at a different time. And so that's a big part of the project. But just to let you know a little bit more about like the workplace navigation and how we see these skills, which I'll talk about in just a second, as being distinct from the other pieces of a really important biotech program or career training education experience, right? We are expected constantly, and rightly so, to go to our industry partners and ask what they want, right? What skills do they need? What employability, like technical and employability skills do our students need? And that is right. We should absolutely produce that and make sure that our students have those skills. But on the opposite side, our students are each individuals of their own. They have goals and priorities and values that are specific to them. And they need ways to make sure that they're navigating that path in a way that is going to make sure that they succeed, that they persist, and they progress in that field. Because we are putting a ton of effort in making sure that they have all of these skills. We want them to stay in that career path. That's the whole point, right? And to succeed in that career path. And so we started to identify skills that we felt were in alignment with trying to make sure students had that capacity. This goes into 3 fields. I have Nalidi, if you have a second, if you drop the link there that's on the slide. If you guys, we have a little form that you can see all of these lists of skills if you'd like. But really came across in 3 areas. One, workplace selection. So students thinking about how they're choosing what workplace they want to be in, or at least being clear about assessing the opportunities that they have and designing interview questions to determine whether or not, like gather the data they need to find out if an opportunity is in alignment with what they want and what they need. Transition, once they start in a new place, how are they establishing themselves? How are they assessing the environment? How are they discerning what's expected of them? How are they making sure they get clarity around that and the feedback they need to move forward effectively? How can they have challenging conversations with people with greater authority than them, especially their supervisors? How are they building their network? Then as they're thinking about progressing, because obviously all of our students are moving on to, throughout their career, they'll move to multiple places, but usually we're doing an internship, they're going to move into a new job. They then really not only thinking about how they articulate the experience they had, but how are they thinking reflectively about that experience? What structures and practices during that last experience both support their goals and sort of and hinder their success, so that they can even come back all the way around and make better choices the next time. So they're constantly moving more forward to something that's going to help them succeed in what they want. So those skills we've predominantly packaged into 4 workshops: utilizing an interview to determine a good fit, establishing yourself in a new position, managing expectations, and negotiating for what you need. We're now on like the 8th iteration of those. They've kind of come together, especially in the last 4 years. And I wanted to give you a little bit of sense of impacts, and then we'll talk about the training that's coming up. Having gathered data from about the last 4 years, we've seen really high levels of trust in our students for their supervisor. Like they're trusting their supervisors much more. They now, the vast majority, feel they can share difficult information with their supervisors. 100% of them now understand what's expected of them. In return, our supervisors are they're trusting their interns at a fairly high level. 97% of them said that they would give a positive recommendation to their intern, and 94% are interested in hosting future community college students. And we have a lot of repeat mentors now. So it's been great. There's been a lot of positive. There's always new things that come up, but it's been a really positive change. And for me, I just want to say, especially if there's any other work-based learning coordinators that are out there and you're trying to get students in internships or work them through those things, things that have been great is that in general, I have fewer internship challenges because students are managing those initially on the front end. They have some skills and tools to do that. There's structures and tools for me to systematically engage students. So when challenges do happen, I'm not like, oh man, I just hope this semester gets over and we'll make sure it doesn't happen next time. There's sort of ways for me to progress forward. It has led to reduced time and effort and stress on my side, and increased confidence and just sustainability of being able to continue to manage a work-based learning program, and more host sites, definitely many more host sites. We usually have more than we have students. Additionally, 95% of our alumni that have gone through the program have said that these skills in particular are critical or very important to their success in the workplace. I won't read the quote just for timing, but it's been that has been sort of the most reassuring part for me, right? Like they feel that this is something that they need. Just to let you know, if you haven't been to one of our workshops before, these are interactive workshops on Zoom. They include breakout discussions, polls, lots of application to the tools that are provided, case studies, or to their students' own experiences. We're really thinking about application oriented, so practical tools students can put into place while they're in the workplace. So frameworks, example language, things like that. This is the Bioscience Career Preparation Series in which the workplace navigation workshops have been embedded this semester. Huge shout out to Linnea and Cassandra for really making this happen through InnovATEBIO this semester. On top of those 4 workshops, just for those of you who may have not seen it otherwise, Dr. Sandy Porter will give a great presentation also on biotech careers, and we'll be doing a speed networking event for students at the end of that semester as well, or at the end of the series. And now I'm going to have Nalini join in instead, so you don't just hear my voice. Just to say, everybody who's associated with community colleges can attend. You can attend, your colleagues can attend, your students can attend, and if you have alumni that are currently job searching, or even if they're not, they are all welcome to attend. But please send this information out to them if you think it's relevant to them. And then sorry, Nalini. Now I will.
No, that's wonderful. So hello, everyone. My name is Nalini Saul, and I'm the director of the Office of Career and Professional Development at UCSF, and I primarily focus on teaching students professional skills, how to navigate professional environments and relationships. And so this particular session, in fact, all of our sessions sort of operates under this idea that we want students to be strategic. And what that means, it's sort of a nice little kind of mnemonic, which is CASA, which is that we want students to be able to walk into an environment and be able to sort of center themselves, to assess their environment and themselves, to be strategic, and then to act. Right? So center, assess, strategize, act. And so every single session that we're doing really focuses on helping students figure out sort of what they should be paying attention to and how to assess, and then how to really reach out to their community, mentors, coordinators, intern coordinators, their friends, to strategize whatever situation they're facing, and then to sort of iteratively act, and sort of take in data to sort of see if it's working or not working. This particular session is one that is frequently an issue, not obviously for every single person, which is, I'm sure you might have been in a situation where you have interviewed at a place and you thought it was great, and then you took the opportunity and then you realized it wasn't, and you're kind of wondering sort of what you missed. And so this session really drills down on this on sort of the three problems that we were seeing. One was that there was a group of students who were interviewing and entirely missed red flags, and red flags being, and not necessarily that the lab was toxic, but it just wasn't going to be a good fit for them. There was a second group and so they missed it. There's a second group that actually kind of saw the issues, but they didn't recognize them as being issues. And so the example that I usually give in the training is that there was a student I once worked with who interviewed, and her interviewer was very polite, very engaged. And then her interviewer sort of heard something going on outside. They got up, they went outside, and they yelled at someone. We don't know who. And then they came back in and were very calm again. And the person was like, I mean, is that an issue? I mean, everyone has a bad day. I mean, right? And then actually 6 months later, she found out, no, it wasn't a bad day. That was called Tuesday for that person. And in fact, they did a lot. So there's the second group, which is that they see issues that actually say that it might be toxic or a bad fit, and they don't recognize it. And then there's a third group that sees it, they recognize it, and then they talk themselves into it. Like, I've right? Like these people are saying that they need a self-starter. What they really mean is that they're going to give me no training, but they say self-starter and I'm a self-starter. And so they talk themselves into environments that are not a good fit. So this session is designed to teach students how to kind of recognize issues, how to assess them, and really how to deal with them. And as Karen is saying, actually every single session really focuses on having people kind of latch on to a framework of assessment, because one of the things we found was that even when people kind of recognized it, they were hesitant to even talk to anyone about it because they didn't even have the language, right? How do I say, my person seems to be really like dismissive of me. And so we also kind of focus on this idea that says that once you can assess it, part of acting is being able to have the language. And so as Karen has said, that I think this is my assessment. I think that people have such a positive outcome to this because it works on the idea that sort of says that when something happens, there's something you can do. Right? Like this is you taking agency. This is you engaging in assessment. This is you engaging in strategy. This is you engaging in acting, rather than just sort of feeling overwhelmed where you're like, I've got no choices, I've got no options, I just need to push through. And the final thing I'll say about this is that this framework, or these frameworks actually are kind of based on something that's adjacent, which is that I used to work at Amherst College, and at the time I was the study abroad advisor when I was in the Career Center. And study abroad operates under the principle that you are going to allow a student to go to an entirely another country and you will not be there. And you need to think about what's the base information that they need to establish themselves, to recognize issues, and then to move, and then to kind of figure out how to navigate it. And so you're kind of always sort of preparing a person, sort of like a scout, right, to be ready for this other experience. I will say that I am super happy that people are that we're really focusing on this idea that returns agency to the student, and also hopefully gives them language to really talk to their circle of support. So that's what I'll just say about the training. But we hope you can come or send your people.
Thanks, Nalini. I think the one piece I want to add on that is I know that sometimes we've gotten faculty who are like, but this is the only opportunity my student will have. I actually only have 3 spots and I got 5 students. So whatever they get is what they're going to get. And still the assessment is really important. If your student's going to walk into a situation is not going to be a great fit for them, they are going to do much better if they're completely aware of what the issues are. And then they can be like, this is why I'm here. It's going to be for this amount of time. And I will be looking for something different for my next opportunity. So,
Right.
and yeah.
And we do talk about that, right? Like the idea isn't that there are times when actually you being there is still in alignment with your goals. It's just going to be more challenging. And the main thing is that you just need to be more proactive, right? So it's not, oh, I had a disagreement with my PI today or with my advisor or my mentor today. I guess it's fine. It's, oh, I had a disagreement with my PI. I should probably check in with my intern coordinator and just sort of tell them about the situation because you're sort of on high alert. And then you are proactively engaging in strategies. So the idea isn't that it sort of takes you out of the game. It's more that you lean into strategy. And I think that for some students and actually, sorry, I work postdocs too, with some students, they don't necessarily, it hasn't been explained to them what that is. And so they just kind of hunker down and try to endure and kind of keep their head down as compared to being strategic. And that's the difference that we're trying to get to.
Yeah. All right, I know I'm running out of time, so I'm going to very quickly, because I know Joseph has a great presentation to give, so I'll go super quickly. I just want to make sure you guys had an overview of the training. We are doing other things in this project besides just the Zoom workshops. We are building a workplace navigation hub with InnovATEBIO. Again, we're going to be converting those Zoom trainings into Canvas, and we're developing communities of practice around workplace navigation and work-based learning. If any of that is of interest to you, I really hope you'll reach out. We'd love to hear from you. Additionally, just to let you know, the Canvas modules are being built right now. They're being piloted with 15 community college focused programs. They should be available kind of more broadly for people to start piloting in fall. So if you are interested, let me know. There are also other ways to utilize Canvas, even if you don't have Canvas. So if this still looks of interest, please reach out. And with that, just a reminder, starting next week on Friday will be the first of these workshops that'll be available. The Workplace Navigation Hub will be coming later this spring, and then the Canvas modules coming in fall. And yeah, I hope to hear from people and look forward to Joseph's presentation.
Thank you, Karen, and also Naladi for joining us. Our second Project Talk speakers are led by Dr. Joseph Olenichak. This project expands workforce training in nanopore sequencing to prepare students for careers in the U.S. bioscience industry. Joe?
Hey, thank you very much for having us, and thank you to InnovATEBIO for putting on this ATE project talk series. My group is really excited to share what we've been working on. We started our NSF ATE project in August of 25, so we're about halfway through year 1. And specifically, we're developing some mini cures and micro-credentials for students that are nanopore based. We have a project team that is nationwide. Myself, Joseph Olenichak here at Austin Community College, Anna Feitzinger from Cold Spring Harbor Laboratory DNA Learning Center, Kasia Dinkaloo from the University of Texas at Austin, Savannah Sin from Los Angeles Pierce College, and Angela Kansani from the Bioscience Core Skills Institute. Our project really has 4 major goals. Our first goal is to develop 2 nanopore sequencing-based mini-cures. Our second goal is to develop 2 micro-credentials that can certify nanopore sequencing skills that students learn as part of these mini-cures. And in this year 1, that's a majority of our work is just developing those products. Next year, after we've developed all of those, we plan to pilot those with students and then iterate them based on student feedback, instructor feedback, as well as industry feedback. And then lastly, in the final year of our project, we hope to disseminate this to other programs that are interested in adopting the resources that we develop. Very briefly, we're going to talk more specifically about the mini cures and micro-credentials. Most of the audience here is probably familiar with cures, these course undergraduate research experiences, which are usually a semester long or multiple semesters long. Really, the idea here for mini cures is developing something much smaller that can be embedded within a course, or maybe something that you can do during an intersession or during a summer session. So these are 4 to 6 week research experiences. They're all nanopore based sequencing, and they involve metabarcoding. The other unique thing about the mini cures we're developing is we're making them scalable so that you can use them in a high school classroom, a 2-year setting, as well as in a 4-year setting. The micro credentials that we're developing are also nanopore based, and these are intended to work with these mini cures so that after a student goes through the mini cure experience, he or she could test for these micro credentials and get an industry certification that shows that they know how to do nanopore sequencing. So at this point, I'd like to pass it off to my co-principal investigator, Anna Feitzinger, to talk a little bit more about nanopore sequencing and specifically the sequencing chemistries we're using.
All right. So just a bit about nanopore sequencing, and Kasia and Savannah will talk a little bit more in detail about the actual mini cures that we're developing. But in general, the device itself, if I'm holding it, so I don't know if you can see my little Zoom window, but it fits in the palm of a hand and it's relatively affordable. So the company has special education pricing so that the startup cost is less than $2,000. And this really makes it the first research and industry grade sequencer that can easily be used in the classroom. Briefly, how it works is that DNA goes through a protein nanopore, which disrupts a current across the pore, which is characteristic to the sequence, and that raw electrical signal that is converted to actual DNA sequences. So in the cures, students can extract DNA from either soil or from a gut that's dissected, which will be described shortly, and use a library prep kit to amplify and sequence a full-length 16S region, which is used to identify all of the different bacteria in the sample. So this is looking at microbiome specifically. Currently, students can use a software that is available through the company, Oxford Nanopore Technologies, called Epitome 2 Labs, to determine the relative abundances or relative abundance of different bacteria in their samples. But I will mention that the DNA Learning Center, through another ATE grant, is working on upgrading our website, DNA Subway, which is a bioinformatics platform designed specifically for student and educators to include a bioinformatics workflow specifically for 16S metabarcoding using nanopore sequencing, which will be better suited for training and education. So that should be available in the next few months, and we'll publicize that through InnovATEBIO channels when we finalize that information. So I think I'm going to pass it off now to, I think Kasia's next, which she'll describe some of the mini cures.
Yeah. So, hi everyone. My name is Kasia Dinklu. I'm an assistant professor of practice at University of Texas at Austin, where I run a lab through the Freshman Research Initiative. So I primarily work with undergraduate students in a wet lab to teach them basic research skills and technical skills the first 2 years of their time at UT, and then they can choose to go forward or not. So in my lab, we work on groups have shown that these beetle larvae will actually eat different types of plastic. And further studies showed that that plastic can actually, or those microbes can actually degrade the plastic, the microbes that live in the gut of the superworms. So our cure is based around finding interesting plastic products made from different plastic polymers that the worms are capable of eating. And students complete feeding trials and kind of monitor the worms as they eat the plastic. And at the end of the feeding trial, they can extract DNA either from the worm frass, which is their poop, or from the worm gut directly. And they can use that DNA to analyze the microbiome through that 16S nanopore sequencing. So the first half of the mini cure is running the feeding trials, collecting good data, monitoring the worms, and then the second half is all about preparing the libraries for 16S barcoding, running the sequencing, and then analyzing the data with Epitome or hopefully DNA Subway 2.0 in the near future.
All right, next up from our team is Savannah.
Yes, thank you. I'm Savannah Sen from LA Pierce College, and our work with the soils minicure is focusing on the soils and ag genetics classes, and we're creating authentic undergraduate research experiences. We've developed some modules using real data consisting of lecture slides and bioinformatics exercises. And you can see students taking samples here out in the field are pre-piloting, where we've gotten our preliminary data. We've been working with strawberries, and students are looking into decreasing dependency on plastic in agriculture, comparing plastic with different types of cover and bare soil. Can go ahead and advance. And it turns out that plastic has a lot of biological and chemical effects on the soil, one of which is inhibiting phosphorus uptake by the plant roots, which of course are closely associated with the microbiome. And one of the interesting results that we put into the module from our data is that the plastics tended to have one of the lowest available phosphorus in the soil compared to the other treatments. You can go ahead and advance. So we're in these modules, we're also including some of the ecological background to make the data more interpretable for students. So for our Nanopore DNA sequencing, they've used Epitome to look at the abundance and composition, and also some downstream tools, and then looking at the alpha diversity. One of the interesting results that they found is that the plastic treatment had one of the highest bacterial species number of sequences detected, which makes sense since plastic is such a good substrate for bacteria to grow on. So we're excited about the pre-piloting and moving into piloting with the whole class starting in next semester and next year. Thank you. And I'll turn it over to Angela Consani.
Hello, everybody. I'm Angela Consani. I'm the CEO and co-founder of the Bioscience Core Skills Institute. For those of you who don't know what we do develop skills validation tests. And when students can pass our tests, we award them a digital micro-credential as evidence for what they can actually do. So our role on the grant is the development of 2 new micro-credentials around nanopore sequencing. So a lot of times I get asked, why are we doing this? So why Nanopore needs skills validation? We now know that Nanopore platforms are being used in a lot of different laboratory environments, but technician readiness varies wildly. This causes employer frustration because they are unable to consistently predict for outcomes in their hiring. So by awarding these evidence-based micro-credentials, this allows employers to have no uncertainty around what these entry-level technicians will actually be able to do in relation to Nanopore systems. So really excited about this project. Next slide. So what we've done so far, when BCSI builds a new micro-credential, we always start with the employers. We realize that it feels good to talk about how we're serving students, which we absolutely are. But in order to serve students, we must serve the employers first and gain their trust. And the way that we do that is by working very closely with them on the development of these skills tests. So the first step in this process, which we've been working on now for the last 6 months, Joe assembled a really high quality team in the form of a BILT, or a business and industry leadership team, for us to query through a series of survey instruments as to what skills they found would be the most exciting to see on a resume in a young technician as it relates to nanopore sequencing. We always ask our employers to stick to those skills that are measurable, trainable, and observable so that we can design a test around them. After going through 2 rounds of survey work where they rank the skill or based on various criteria, such as the trainability or the cost of training, things like that, they did determine that these are the top 4 skills that they would see as the prioritized skills that matter the most on the job: sample preparation, flow cell setup, run monitoring, basic troubleshooting, and then the data awareness piece. So we're still deciding, and the team is now voting on which of these skills will be the final 2, and then we'll set to work on the test design. Next slide. So how we validate, this is hands-on performance-based testing. The students will be expected to perform the skill in front of someone who has been certified and trained in our rubrics. And if the student is able to pass the performance-based test, then we will award them a digital micro-credential that they can use as evidence that they have been trained differently than the average student and they do have the skills. Dave, I saw your question in the chat, and that is what the grant allowed. So we had room for 2. We didn't want to overstress ourselves in these 3 years while also developing the mini cures. Next slide. So why does this matter to InnovATEBIO? Needless to say, it establishes a replicable model for validating these emerging lab technologies. It also, and this is the key piece, it garners the employer trust for InnovATEBIO training programs in that you're graduating individuals who have been trained differently than the average student, and they should be able to convey that into their jobs. We're going to collect a lot of data around that. We already collected some fascinating data from the employers, and it positions InnovATEBIO as a leader in skills-first workforce infrastructure. And with that, I'll turn it over to Joe.
All righty. Like we mentioned at the beginning of this talk, we are 6 months into our first year on a 3-year project. So moving forward for the remainder of this semester, our group finishes up our work developing these 2 mini cures, as well as developing these 2 micro-credentials and testing those micro-credentials. In the fall, we plan to actually pilot this with students. So we've identified some high school sites, as well as some community college sites, as well as a 4-year site. We'll get some feedback from students, as well as instructors and industry. We'll iterate those mini cures, as well as update those micro-credentials. And then moving into the last year of the grant, we hope to disseminate it to other partners here through the InnovATEBIO network and community colleges, high school programs, and 4-year programs in general to take these mini cures and implement them at their institutions, as well as these micro-credentials. And with that, I will end our talk.
Thank you so much for the second Project Talk speakers, a great group. We now have time for a Q&A session for both of our teams who presented today. I do see some questions in the chat. Angela, you addressed the first one, but Linnea, you asked a question regarding the plastic of function of both worm and bacteria. Do you want to go ahead and address that?
She answered the question. My only question
was,
I gather, does it require that the worm is digesting it and then the bacteria digest it, or is it just the fact that the plastic is in the gut and then the bacteria digest the plastic? Because is it synergistic that it takes both? But I think you said that it doesn't.
So in a sense that a lot of the gut microbes, like that's a very niche environment. So they're typically hard to culture away from the worm. So they exist in the gut, and the challenge is always finding a way to get the plastic to them inside of this incubator. So worm ingestion is one part of the problem, but as far as like digestion versus depolymerization, we monitor the worm frass to make sure there are plastic fragments in it. because that's how we confirm that it
It's
passed
just like a
through.
little incubator then.
Yes,
It's the gut is
exactly
the
that.
incubator. So
Yep.
it provides, the right conditions, but does it take synergy between the bacteria too, such that one bacteria degrades part of it and then another bacteria? So it takes that whole community to make this work. So if you look at the enzymatic process, it's the overlap between the several of the bacteria to make it happen. My husband worked on creation of the cochineal dye, and so that took a gut microbiome and the fact that they were in a plant to do this.
Yes, yeah, and it's
I'm
definitely
saying,
like polymer by polymer basis and microbiome by microbiome basis. So sometimes we're able to isolate a single species that's depolymerizing several steps
okay,
versus needing the consortium of microbes where everybody's doing their part to break down the polymer chain. Uh,
cool.
yeah, so the first step is always getting that good metagenomic data, and then there's a lot of downstream analysis when we start
Yeah, which enzymes
isolating.
are doing what? Yeah,
Yes, exactly.
cool.
Anjali had a question in the chat. Do you want to raise that?
Yeah, hi everyone. My question is about the sequencing data that students collected from nanopore sequencing. Um, and one of the things that nanopore sequencing can also be used is to identify RNA modifications. So, did students looked into that as well when they were collecting that data?
We have not done any work with RNA in the nanopore. It's all been DNA based so far.
Okay, sorry, I was not sure if it was like it included RNA analysis or not.
I have a question for Karen. I didn't write it in chat though. So on the career navigation, I'm going to be really interested in learning the impact even on alumni, because that is one group I'm really interested in taking advantage of this, because we have never bothered. That is not quite the right word to say, bothered. We have not trained alumni in the workplace, okay? We've always done it with our students, and now we're going to offer this for our alumni. Do you have some specific case studies about how that has made a difference?
So I'll say I'm very excited about this being offered to alumni, that in some of these things, this is not a great analogy, but I think Naledi is really good at saying, like, this is like the things that we train in, or that Naledi specifically trains in the workplace navigation, is almost like doing your taxes. You don't learn how to do your taxes until, right? You don't learn how to negotiate a conversation with your boss until you're about to have a really bad conversation that you need that, you need that skill. And so actually that session, that last workshop, came because we were talking to our alumni about what they needed while they were in the workplace. What was the thing that was still missing? So just to say, I think yeah, I'm very excited about them being able to have access to it when they
Well, the
need it.
hands that went up over that. So, because to me,
Oh, they were there before.
they were there before. I wish someone had done that with me, because I could have really used that in negotiating things with my advisor, to be quite honest, and then my postdoc advisors.
Right. I think the only thing I can say is that since I primarily work with graduate students and postdoctoral scholars, I know that this training works with them because I teach them. And I also know we also see alumni up to a year after they leave. And I have had conversations with students and postdocs who have said, I actually didn't pay attention to what you said. I took the job, and then now I'm in the situation, and well, they can see us up to a year later. And so now I'd like to come back and actually try it. But the idea is that the ability to actually sort of quote unquote take charge of your career, your ability to assess and navigate workspaces and environments are lifelong skills. They don't necessarily change, they just sort of sometimes get a little bit more complex.
Thank you. We have a hand up from Thomas.
Yes, so I have a question and then a shameless plug for a possible local neighbor New Yorker collaboration with Anna and the DNA crew for Nanopore. So my question is with the Nanopore. So the squiggles that you get when the DNA is coming down the membrane, right, with the change in the electrical current, is that basically as each nucleotide is going, you get the squiggle for that long piece that's being read?
It's not like one base has a corresponding peak in the electric in that current change over time. It's actually that a chunk of sequence is a pattern that's being recognized using the neural network algorithms that processes those data. So it's really pattern recognition is what's going on.
But you measure the electrical change across the membrane potential. That's being read as very raw data that's fed into the neural network.
Yeah, so it's essentially a squiggle graph, and that data, the algorithms that process that are pattern recognition algorithms. So it's not it's getting base called chunks of DNA, like 4 to 5 base pairs at a time.
So
That
now
also ex
here comes my shameless neighborly Mr. Rogers neighborhood plug of will you be my neighbor in terms of mini cure type stuff. Because I was just speaking with Allison for the urban barcoding research program, a mentor for students. We're doing a project, and when I was talking, and then Sandra and others might know that I'm into data sonification and linking music and molecular biology, cell biology. And I was chatting with Allison quickly about the nanopore for that work. And then I was thinking, and she triggered me on to something, is possibly maybe doing either for this summer a potential mini cure for our QSTEM program at LaGuardia, but also to looking at how we could even do with non-majors and interdisciplinary work for looking at not just looking at like the sequencing part, but on how you could develop and integrate sonification into your nanopore technology, where as the raw data is coming, can we have algorithms that are recognizing music patterns to put that to sound? So now you're hearing the DNA as it's being sequenced, and you could identify patterns that way in addition to the regular based out reading that you're getting. So I'm really interested in possibly talking more with you and your group in terms of how we could use this to integrate data sonification and music incorporation into nanopore sequencing and possibly mini cure experiences for both non-majors and majors in biotech.
Yeah, I'm happy to talk more. Allison told me a bit about that actually. She mentioned your conversation. I'm also a music person, so
Perfect.
we can talk if you want to email me. I'll put my email in the
I
chat.
have it already. So yes, I
Oh,
have it.
perfect.
Yes,
So
yes,
yeah,
yes.
reach out anytime. Actually,
Okay.
Allison was just at LaGuardia this morning doing bioskill evaluations for some of the faculty. So awesome connections.
Perfect. Thank
I think Dave is going to mention one thing. I saw Dave unmuted when were you going to mention something corresponding to the
Oh
base
yeah, I was
call?
just going to say when Tom was mentioning that uh, that, and I forgot exactly now the context, but that decoding of the so-called squiggles of the primary data into sequence is computationally intensive and typically is best done on GPUs. So if you also include layer music on there, it would probably be quite computationally intensive, but that doesn't mean it shouldn't be done.
But we can talk. I have ideas. We can talk offline.
Thank you for your question, Tom. Maggie, you have your hand up?
Yeah, my question is for Karen. Karen, can you talk a little bit more about your synergy or connection with your four-year partner, UCSF? And I'm asking because my four-year partner, Scott Gigax, is on this call at Thomas Jefferson University, and we work very closely together with our transfer students.
Yeah, definitely. And surprisingly enough, so actually UCSF is not a four-year institution. It is only a graduate and medical school. And Nalini can probably talk about the pharmacy school. It's a very interesting partnership because they don't have an undergraduate population. So the majority of our interns go do internships there, and it's very successful in that manner. It's been a fantastic partnership. To a certain extent, we had, so when I was talking originally, Laurence Clement, who had worked with James and also Ying Su in developing the program originally, had moved to the Office of Career and Professional Development and worked with Naliti. And so we had this kind of this cross-pollination. It's been, and then I would love to have Nalini say something, but it's been extremely useful to have people on either side, right? Like we're working with the students, and then, and then the mentors are working, internship mentors are working with the students, but you get to see the challenges on both sides and thinking about what will work for both of them.
Right. And initially, there was actually a much more intense program that included there being mentors, including students and postdocs and faculty from UCSF being mentors to the City College students. And so it was a great opportunity for the mentees to learn and the mentors to learn, and then to practice with each other in a dyad with us coaching them. Right. And so for our students and for the students and postdocs at UCSF, it was one of the first times that they had a chance to learn people skills, even as a research mentor. How do I set expectations? How do I give feedback? And then they could also, since they were also trainees, right? They're then also thinking about this sort of their relationship, both from the perspective of being a mentor and being a mentee. So it gave them a really unique opportunity to kind of practice both sides of the coin. And we did find that that the mentors in industry, faculty at UCSF, they were excited because in many cases they hadn't ever received people manager training. And so the chance to sort of practice with an intern who was trained in a very low stakes way was one of the things that we think actually kind of increased the number of mentor participants and got us to the point where we had more mentors than mentees.
Yeah.
Right. Thanks.
Thank you, Maggie. Scott, you have your hand up.
Yes, I appreciated both talks. I thought it was excellent, and I'm a relative newcomer to the group, so I'm so happy to be here. Like Maggie was saying, I work very closely with Maggie. One of the things that we do is have relationships and partners with industry as far as understanding where our students would graduate and where they would be employed. When you reach out to industry members, are there certain industry members that are more apt to take students at different levels as far as the associates or bachelor's level in your different areas? Like, are there kind of skill sets that you see kind of across the board that we should be focused on?
Yeah, that's a very, I'm guessing the question's to me, Scott.
Yeah, sorry.
Okay, that's all right. So it is highly variable because the Bay Area, we have something like over 2,000, San Francisco Bay Area, over 2,000 employers. So we rarely actually even have UCSF labs necessarily take an intern. We do, but postdocs are cyclical and they will take them for a period of time. I've been really interested in the fact that different mentors are just very interested, different hosts are very interested in different skill levels. One of the things we do now when we have mentors that are interested is we very clearly have them outline what sort of like their base level of skill that they would want. And we get people who are just like, I just want people who are motivated and excited about the things that I'm excited about, to people who are like, I need this base skill level. They need to absolutely know aseptic technique, but or they need to know cell culture to a certain level. Part of the strategy, which does come, it's not necessarily the workplace navigation training, but in structurally setting up that program, is defining that for mentors ahead of time and making sure that information is shared with students so they are making strategic moves about where they should reach out. We definitely have had students that have very little skill level, sort of like in a lab sense, but they actually have a lot of work experience. So they actually are really good at going in and actually working with a team and working with maybe not the difficult personalities or right kind of navigating space, and they can be extremely effective in their internship. There's a lot of, I would be happy to have like further conversations. I would actually really be excited to have those conversations because I think there's a lot of discussion to make that work in a way that everybody's on board and at the same point and is happy to navigate that. Yeah.
Right. Yeah, that'd be great because us being in the Philadelphia region, we have our kind of own industry cluster, and be kind of interested to see the comparisons.
Yeah,
Thank you, Karen.
definitely. Yeah.
Thank you, Scott. Does anyone have any other questions for our speakers or comments?
Can I ask one quick, although it's kind of been answered? I'm really interested, Karen, in sort of the different career trajectories of students that have had the workplace navigation training versus students who never did. And if you, I know you haven't been collecting data that long, but do you have anecdotes or have you heard things?
It's wrapped in so many other pieces.
Yeah.
Yeah, I feel like students now more are, at least, maybe I'm not. Sorry, yeah, I think that it's like so qualitative, not quantitative in any sense of them going where they go. A lot of them are being much more thoughtful, I think, about trying to determine. So one of the things we have is, like, because we have academic labs and government labs and companies, right, is students kind of trying to decide which one of those types of environments is interesting to them. And we do have more this isn't getting to your answer, but more conversations about whether that particular experience was what they wanted, right? Did they like being in industry? Or a lot of it's academic labs. Did they like being in that sort of, tends to be a little less structured, right? Kind of just generally, we're going and looking for things versus I want to know how to do a technique that I can do repeatedly over and over again and feel super confident with. That stratification comes more there, and talking to other students about that, and actually doing the speed networking and alumni coming back and being like, oh, well, actually, if you want to feel really confident, you should go to manufacturing for a while, because they're going to just, like, right, you're going to get really trained. And then I've had students come back and then be like, okay, once I'm there, now I need to go back to research because I'm kind of bored in this space and I want to do something that's more, that's not all students, but those are the things I've heard.
Thanks. No, I find that interesting, and I can see where being more intentional is going to be helpful. I'm also wondering too if it cushions you a little bit better in times where there are a lot of layoffs. But
yeah,
that's, you know.
I mean, we definitely have had alumni that have been laid off in the last couple of years, but they seem to be seeking community pretty regularly, which is great. We hear from them, they reach back out to us. Yeah.
Thanks.
Thank you, Sandy, for your question. And so at this time, we are down to the last minute. I would like to take this time to thank both of our presenters and your teams for joining us. And thank you all to, thanks to everyone else for joining us and for helping create an engaging question and answer session. So as a reminder, the survey link is listed in the chat. We would appreciate we would appreciate you completing that. Thank you. And also the recordings of both presentations will be on the InnovATEBIO website by the end of next week. You can find more, as I mentioned earlier, about these two projects on the InnovATEBIO website, as well as a registration link for Karen's Bioscience Career Preparation Series. It's the link to that is also in the chat at the beginning of her discussion, and it's also on the InnovATEBIO website. And again, we thank you for attending, and we hope to see you next week and for our other Project Talk series. We wish that you all have a great weekend. And Linnea, you have your hand up. You want to say a closing remark?
I just want to remind people, so remember in the ATE solicitation, there was a section that Celeste wrote. And by the way, that solicitation is not going to be changed for, hopefully, as Celeste has advised Olga not to do that. But I wanted to point out, it said in there that if you see a project that is of interest to you, ATE will fund the same project, of course, with nuances for your applications. And people should realize when they watch these ATE talks, this also could be the basis of another grant. So don't forget that.